Hello students, welcome to Read Med Prep Academy channel. In genetics, today we are going to talk about the principles of inheritance and variation part 2. In this we are going to discuss about sex determination in general, sex determination in human beings, bark body, haplodiploidy in honeybees. Regarding the sex determination in general, what is meant by sex determination? Sex determination is a method by which the distinction between the male and the female is established in a species. What determines the sex? The sex chromosomes determine the sex of the individual in a dioecious or a unisexual organism. These sex chromosomes are called allosomes. What are autosomes? The chromosomes other than the sex chromosomes of an individual are called autosomes. You can see here that a human chromosome karyotype consists of 46 chromosomes, that is 23 pairs, out of which 22 pairs are called autosomes, and one pair, two sex chromosomes are present. In the males, it is XY, and in the females, it is XX. What are the types of sex chromosomes? Sex chromosomes may be similar, homomorphic in one sex. Example, in human females, it is XX. Dissimilar, otherwise called heteromorphic in the other, like the human male, which is designated as XY. Here you can see X chromosome and the X chromosome in the female, and the X chromosome and the Y chromosome in a male. Now coming to the gamete production, individuals having homomorphic sex chromosomes produce only one type of gametes. They are called homogamitic, whereas heteromorphic individuals produce two types of gametes, otherwise called heterogamatic. See here that the father has X and a Y chromosome, produces sperms, one type having X chromosome, the other type having Y chromosome. The mother, which has two X chromosomes, produces ova containing only X chromosomes. Hence, the males are heterogamatic and the females are homogamic. What is the size of the allosomes? The human Y chromosome is only 60 MB in size, with 60 to 200 functional genes. X chromosomes are 165 MB, that means they are larger than the Y chromosome in size and contains about 800 to 1000 genes. Here you can see that a X chromosome is larger and a Y chromosome has a sex determining region called SRY gene. What is the chromosomal basis of sex determination? Now let us discuss about the heterogamatic sex determination. In heterogamatic sex determination, one of the sexes produces similar gametes, the other sex produces dissimilar gametes. Sex offspring is determined at the time of fertilization. Two types of heterogamatic sex determination. One is called heterogamatic males, the other one is called heterogamatic females. Now regarding the heterogamatic males, in this method of sex determination, the males are heterogamatic, producing dissimilar gametes, whereas females are homogamic, producing similar gametes. Types of heterogamatic males. There are two kinds of heterogamatic males. XXXO type, which is seen in bugs, cockroaches and grasshoppers. XXXY type, which is seen in human beings and Drosophila. Regarding the XXXO type, the diploid nuclei in the female has two X chromosomes, that is X and X. The male has only one X chromosome. Now the gametes produced by the females are X and X, that means they are homogamic, whereas the males have only one X chromosome in a gamete, that the other does not contain the X chromosome. And by fertilization, the zygotes that are formed contain X and X chromosome, which is a female, and the X and 0 chromosome, which is a male. XXXO system is seen in plants like Vallisneria, insects of the order of Hemiptera and Orthoptera. Females have two X chromosomes and the males have only one X chromosomes. Male produces two types of sperms, one with X chromosome, the other without the X chromosome. The presence of one unpaired X chromosome determines the male sex. So the males will have 22 autosomes plus one X chromosome, whereas the females will have 22 autosomes and two X chromosomes. Now coming to the XXXY system, which is seen in human beings, the parents, the father will have X and Y chromosome, and the gametes that are produced contain one sperm containing X chromosome, the other containing the Y chromosome, whereas the mother contains X chromosome and the ova that is produced will have only one type of chromosome, that is the X chromosome. The zygote that is formed by the fusion of the gametes will have a probability of 50% female child and the probability of 50% male child by fertilization. Now going to heterogamatic females, in this method of sex determination, the females are heterogamatic, producing dissimilar gametes, while the males are homogamatic, producing only one type of gametes. Alphabets used in heterogamatic females. To avoid the confusion with the XXXO and XXXY type of sex determination, the alphabets here used are Z instead of X and W instead of Y are used. The types of heterogamatic females are two types. ZOZZ type, which is seen in moths, butterflies and domestic chickens, and ZWZZ type, which is seen in gypsy moth, fishes, reptiles and birds. ZZZO system. The males will have autosomes plus two Z chromosomes, that is Z and Z, and they produce gametes which contain autosomes and one Z chromosome. Gametes are homogamatic, whereas the females will have autosomes and only one Z chromosome, and they produce gametes which have autosomes along with only one Z chromosome or autosomes without any Z chromosome. And the progeny that is formed will have males containing autosomes and two Z chromosomes and females having autosomes with only one Z chromosome. This is called ZZZO system. In ZZZW system, males will have autosomes and two Z chromosomes, and they produce gametes, autosomes with one Z chromosome, which are homogamatic, whereas in females the autosomes have in addition the ZW chromosome, and the gametes that are produced are autosomes with a Z chromosome and an autosome with a W chromosome. And the progeny that is formed, the males will have autosomes with two Z chromosomes which are homogamatic, and the females will have autosomes with ZW chromosome which will have heterogamatic. Now ZZZW sex determination is heterogamatic females. Males which have ZZ are homogamatic. This type of sex determination is found in birds, snakes, butterflies and some amphibians and some fishes. Here you can see the differences between the heterogamatic males and the heterogamatic females. The heterogamatic males will have subtypes XXXO. The sex chromosomes, the females will have two X chromosome and the males will have one X chromosome. This type of XXXO sex determination is seen in plants like Vallisneria spiralis, bugs and grasshoppers. The subtype XXXY system, the females will have two X chromosomes and the males will have XY chromosome. This type of system is seen in mans and other mammals, Drosophila and certain angiosperms. In heterogamatic females, the subtypes is ZOZZ system. The females will have one Z chromosome and the males will have two Z chromosome. This is seen in moths and butterflies. In subtype ZWZZ system, the females will have ZW and the males will have two Z chromosomes. This type of system is seen in gypsy moths, fishes, reptiles, birds and mammals. Now let us move on to the sex determination in human beings. Genes determining the sex in human beings are located on two sex chromosomes. These sex chromosomes are called allosomes. In mammals, sex determination is associated with chromosomal differences between the two sexes. Typically, XX is present in females and XY is present in males. Here you can see the X chromosomes, which are two in number in the females, which are similar, homologous, whereas in the males the X chromosome is larger and the Y chromosome is smaller. What are the total number of chromosomes that are present in human beings? There are 46 chromosomes, 22 pairs are autosomes and one pair is sex chromosome. The sex chromosomes are XX or XY. This you have already seen earlier. that there are 22 pairs of autosomes and 2 sex chromosomes, XY in males and XX in females. Now something about the discovery of the allosomes. X chromosome was discovered by Henking in 1891. Y chromosome was discovered by Stevens in 1902. What are the types of gametes that are produced in human beings? The females are homogamatic, producing only one type of egg. Each egg contains only one chromosome of X, whereas males are heterogamatic, producing two types of sperms, one with X chromosome, the other with the Y chromosome. Here you can see that the male having the X and Y chromosome will produce a gamete, which is the sperm which has X chromosome or Y chromosome, whereas the mother or the female which has double X chromosome in a diploid cell will produce a haploid gamete of each containing X chromosome. Now, example of organisms having this XX-XY system of chromosomes is seen in Drosophila. Now, here you can see that the heterogamatic males and the homogamatic females, when the gametes are produced, the fertilization offspring has a probability of 50% males and 50% females. The Y chromosome and the male development. Current analysis of Y chromosomes has revealed numerous genes with potential genetic function. Some genes with or without the homologous counterparts are seen on X chromosome. You can see on the right side that the lower part of the X and the Y chromosome are homologous parts, whereas the upper part of the X and the Y chromosome have specific genes which are X-linked in X chromosome and Y-linked in Y chromosome. These parts are non-homologous parts. Present at both ends of the Y chromosome are the pseudo-autosomal regions. These pseudo-autosomal regions are similar on the X chromosome and the Y chromosome, with synapse and recombine during meiosis. Here you can see that the X chromosome is larger and the Y chromosome is smaller, and the terminal ends, the upper part contains the pseudo-autosomal region, which is the primary, and the lower part is the secondary pseudo-autosomal region. These regions are homologous to each other. What is NRY? The remaining 95% of the Y chromosome is referred as the non-combining region of the Y chromosome, or the male-specific region of Y. Is divided equally into functional genes, euchromatic, and non-functional genes, heterochromatic. Here you can see the Y chromosome, which has the pseudo-autosomal regions at both ends, and the euchromatin and the heterochromatin. The Y chromosome spans about 58 million base pairs. It contains over 200 genes, at least 72 of which code for proteins, of which only 17 to 27 were male-specific genes. What is SRY? Within the euchromatin region is a gene called sex-determining region of Y, called SRY, in the short arm. This gene is Y-linked because it is found only in the Y chromosome. What is the function of SRY? In humans, absence of Y chromosome inevitably leads to female development. And this SRY gene is absent in X chromosome. The gene product of SRY is the testis determining factor, TDF, which is present in the male testis. Let us just discuss about the differences between the X and the Y chromosome. X chromosome is longer than the Y chromosome. X chromosome contains large amount of euchromatin and small amount of heterochromatin. The euchromatin contains large amount of DNA or genes. The Y chromosome contains small amount of euchromatin and large amount of heterochromatin. The crossing over between the X and the Y during meiosis occurs only in the pseudo-autosomal region, otherwise PAR, necessary for normal disjunction. PAR homologous to the region of the X chromosome. The non-combining region of Y, NRY, unique to Y chromosome. The human Y chromosome contains genes SRY, which determines the maleness and which makes TDF, testis determining factor. XY females deleted for SRY region are observed, and XX males with TDF gene translocated to Y chromosome are also observed. Now going to Barr body. In 1949, Barr and Bertram first observed a condensed body in the nerve cells of female cat, which was absent in the male. This condensed body was called sex chromatin by them and was later referred as Barr body. Here you can see Murray L. Barr and E.G. Bertram, who discovered the Barr body in 1949. How many X chromosomes are present? In the XY chromosomal system of sex determination, males have only one X chromosome, whereas females have two X chromosomes. Then how does the organism compensate for these dosage differences between the sexes? In mammals, the necessary dosage compensation is accomplished by inactivation of one of the X chromosome in the females, so that both males and the females have only one functional X chromosome per cell. Here you can see that the zygote has two X chromosomes which undergoes random inactivation, where one X chromosome becomes active, the other X chromosome becomes just a body of chromatin, forming a Barr body. And as the mitosis continues, the Barr body also continues in the cell. The Barr body is nothing but the condensed inactive member of a pair of X chromosome in the cell. The other X chromosome is not condensed and is active in transcription. When one Barr body is present in the nucleus, then it indicates a female sex. When there is no Barr body in the nucleus, then it is a male sex. That means when two X chromosomes are present, one Barr body should be present, and when only one X chromosome is present, there is no Barr body. Now, Lyons hypothesis. Mary Frances Lyon suggested that Barr bodies represented an inactive chromosome which in females becomes tightly coiled into a heterochromatin, a condensed and visible form of chromatin. This Lyons hypothesis states that during early development, one of the X chromosomes in a female gets turned off, and this is maintained in all descendant cells of the clone. According to Lyons hypothesis, one X chromosome is generically active in the body cells. The second is inactive and tightly coiled. Either the maternal or the paternal chromosome can be inactivated. Inactivation is permanent, reset in germ cells. Inactivation of second X equalizes the activity of the X-linked genes in males and females. Mary Lyon and others in 2007 was given the Rosenstiel Award for the discovery of this theory. The inactivation of one X chromosome results in the formation of a Barr body and one X chromosome present in all cells of the human beings. What is the rule of the Barr body? The number of Barr bodies observed in a cell was one less than the number of the X chromosome. That the number of Barr bodies follows n-1 rule, where n is the total number of the X chromosomes present. For example, in a male, XY determines that it has no Barr body. XO females have no Barr body since there is only one X chromosome. XXY males have one Barr body. Triple X, when you take triple X, there are three X chromosomes. 3 minus 1, that means there are two Barr bodies. Triple XY, two Barr bodies. Double X, where there are 4 X chromosomes, 4 minus 1 will give 3 Barr bodies. Double XY, that means it has 3 Barr bodies. Here you can see in a normal male there is no Barr body. In a Turner syndrome which has 45 X, there is no Barr body. In a normal female, there is one Barr body. In a Klinefelter syndrome, which is 47 chromosomes, XXY, there are 2 X chromosomes, hence there is only one Barr body. In 47 triple X, there are 2 Barr bodies. In 48 chromosomes, where it has double XXY, it has 2 Barr bodies. In 48 double X, there are 3 Barr bodies. In 49 chromosomes, double XY, it has 3 Barr bodies. Now going to haplodiploidy in honeybees. What is haplodiploidy? In Hymenopteron insects such as honeybees, ants and wasps, a mechanism of sex determination is common. In this system, the sex of the offspring is determined by the number of the sets of the chromosomes it receives. The male and the female honeybees, if you take, the fertilized eggs develop into the females, which are the queens or the workers, and the unfertilized eggs develop into the males, who are the drones by parthenogenesis. Here you can see that the fertilized eggs develops into the females, who is the worker or the queen, and the unfertilized egg develop into a drone. What about the chromosomes in the males and the female honeybees? It means that the males have half the number of chromosomes, that means haploid number of chromosomes. The females have double the number of chromosomes, that means the diploid number of chromosomes. Hence the name haplodiploidy for this system of sex determination. When you take here in this picture a diploid female bee, when it undergoes meiosis, it has two types of eggs. These eggs do not undergo fertilization and they develop into haploid male bees. When you take a diploid female bee, when it undergoes meiosis, it produces haploid number of eggs. And a haploid male bee which does not undergo meiosis and it produces sperms. These haploid sperms and the haploid eggs fuse together to form only diploid female bees by fertilization. This is how the sex is determined in honeybees. Who lays the eggs in honeybees? This mode of sex determination facilitates the evolution of sociality in which only when the one diploid female becomes a queen and only the queen lays the eggs for the colony. Here you can see the drone, which is the male, the queen in the center, which is the female, which only lays the eggs, and the workers which do not lay the egg but they are the females. Here you can see the honeycomb with the eggs. What is kin selection? All other females which are diploid, having developed from fertilized eggs, help to raise the queen's eggs and so contribute to the queen's reproductive success and indirectly to their own. This phenomenon of carrying the queen's eggs is known as kin selection. According to the Hamilton's rule and kin selection, in honeybees, most of the females in a colony do not reproduce. Female workers can increase their overall fitness by caring for the sisters rather than reproducing on their own. So how does the queen control the workers? The queen constructs their social environment by releasing a hormone that suppresses the fertility of the workers. So today we have discussed the sex determination in general, sex determination in human beings, about bar body, haplodiploidy in honeybees. Thank you very much. Kindly subscribe, like, share and comment the channel Read MedPrep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you.